Okay, great. I see it. So, hello everybody. My name is Dr. Vihang Narkar. I am an associate professor at the Institute of Molecular Medicine, which is the part of the medical school. My lab is in the institute. My lab is in the Center for Metabolic and Degenerative Diseases, and I'm also affiliated with the Department of Integrative Biology and Pharmacology. And so if I can have the next slide, there you go. And so the research in my lab is really entirely focused on understanding the molecular basis of exercise adaptations or um, the mechanisms of exercise benefits in diseases. And so as we all know or have been known for a long time, exercise really has fantastic health benefits in a variety of diseases, but particularly in metabolic diseases like diabetes and obesity, as well as in cardiovascular diseases. And so what we are interested in then understanding is what are the signaling pathways or genetic pathways underlying exercise that leads to benefits in these different disease models. And so if we can identify these pathways or specific biomarker genes, then we can directly activate these genes in absence of exercise to induce exercise mimetic effects and prevent disease. And so if you look at panel 2 here, it's a typical gene signature. And again, this is data from mice, which are subjected to exercise training. And this gene signature contains a variety of genes, transcriptional factors, kinases that are activated by exercise. And so we use high throughput genetics and bioinformatic analysis to identify highly likely genes or pathways that can mediate the effects of exercise. If you look at panel 3, our other approaches then to pick selective target genes from such a signature and activate them in what we fondly call as couch potato mice. And so we use typically pharmacology, gene therapy, or genetic engineering to do this and ask a simple question: does activating that particular target pathway leads to a transformation of this couch potato mouse into a marathon mouse. And so we've been doing this now for over several years, and we've had quite a bit of success identifying such genes. And one thing I forgot to mention here is that we're particularly interested in transcriptional factors. And the reason for that is transcriptional factors, if you activate them, can bring about large-scale remodeling in organ systems to kind of mimic exercise. And so if you look down here on panel 5, that's one of our transgenic mouse that we fondly call as marathon mouse. And it's in which a gene called ERR gamma, this is an orphan nuclear receptor, is specifically activated in the skeletal muscle. And you can see that by simply turning on one gene, you can turn a normal mouse into a marathon mouse. This is the red line here indicates the time for which the transgenic mouse runs and the distance for which it runs. And the video here will show you that this guy here is a marathon mouse, and this one is wild type. And if you keep looking at it, you'll see that the transgenic mouse tends to run longer than the normal mouse. And so we don't stop there. We then take these mice, analyze their different tissues. What we find is that, for example, here in the skeletal muscle, you have improved vasculature, increased mitochondrial capacity. And what this suggests is that it's at least in a proof of principle manner that you're simply one gene or one receptor away from mimicking some of the benefits of exercise. We also apply this to disease models. Here is an example of an obese mouse. This is a genetically obese mouse. And if you activate one such exercise mimetic gene, in this case PPR delta, it's also a transcriptional factor. You can see that these guys dramatically lose weight, and really they don't stop eating as much as this particular tubby mouse. And so that's one example, as I said, and we apply this sort of exercise mimetic genes to different disease models. Some other things we're interested in are muscular dystrophy, orphan diseases like Duchenne's, as well as cardiovascular diseases like peripheral arterial disease. And here are some of our publications if you're interested in following them up. But overall, the overarching goal of my lab then is to use exercise physiology as a model system to identify genes in these different organs that could benefit from exercise and go after those genes in absence of exercise and see if we can identify exercise mimetic pathways that can be utilized for therapeutics. And so a student in my lab would be really exposed to training in high throughput genomics, as I said, generation of transgenic mouse lines, gain of function, loss of function, a lot of mechanistic studies with transcriptional factors, as well as cool exercise physiology, as you see in this video, and exploration of disease models, particularly diabetes and obesity. And so thank you for your attention, and I'll be happy to answer any questions.